तो यह है जेनेटिक्स एंड एवोल्यूशन का एनसीईआरटी। एनसीईआरटी कहता है कि the work of the Mendel and the other who followed him gave us an idea of the inheritance pattern. However, the nature of those factors which determine the phenotype are not very clear. As these factors represent the genetic basis of the inheritance, understanding the structure of the genetic material and the structural basis of the genotype and the phenotype conversion become the focus of the attention in the biology for the next century. The entire body of the molecular biology was a consequent development with a major contribution from the Bartson, Crick, Nierenberg, Khurana, Kornberg's father and the son, Benger, Monod, Brenner, etc. A parallel problem being tracked by the mechanism of the evolution, awareness in the areas of the molecular genetics, structural biology, and the bioinformatics have enriched our understanding of the molecular basis of the evolution. In this unit, the structure and the function of the DNA and the story and the theory of the evolution have been examined and explained. फिर आता है फिर कहता है कि जेम्स डेवी वाटसन बाय बॉर्न इन द शिकागो ऑन द 6 अप्रैल 1928 इन द 1947 he received the BSc degree in the zoology. During this year he interested in the bird watching and had matured into serious desire to learn the genetics. This became possible when he received a fellowship for the graduate study in the zoology at the Indiana University, Bloomington, where he received his PhD degree in the 1950 on the study of the effect of the hard X-ray on the bacteriophage multiplication. He met Crick and they discovered their common interest in solving the DNA structure. The first serious effort was unsatisfactory. The second effort, based upon more experimental evidence and better appreciation of the nucleic acid literature, yielded early in the March 1953 in the proposal of the complementary double helical configuration. Francis Harry Compton Crick was born on the 8th June 1916 at the Northampton, England. He studied physics at the University College London and obtained a BSc in the 1937. He completed PhD in 1954 on a thesis entitled X-ray diffraction, polypeptide and the proteins. फिर कहता है कि a critical influence in the Crick career was his friendship with the J.D. Bartson, then a young man of the 23, leading in 1953 to the proposal of the double helical structure for the DNA and the replication scheme. Crick was made on an FRF in 1959. The honors to the Bartson and Crick included John Colin Warren Prize of the Massachusetts General Hospital in 1959, the Lasker Award in 1950, the Research Corporation Prize in 1952, and above all the Nobel Prize in 1952. तो फिर आता है द प्रिंसिपल ऑफ द इनहेरिटेंस एंड द वेरिएशन. इसमें मेंडल्स लॉ ऑफ द इनहेरिटेंस आता है इनहेरिटेंस ऑफ द वन जीन, इनहेरिटेंस ऑफ द टू जीन, सेक्स डिटर्मिनेशन, म्यूटेशन और जेनेटिक डिसऑर्डर्स आते हैं. तो एनसीईआरटी कहता है कि हैव यू एवर बॉन्डेड बाय एन एलिफेंट ऑलवेज गिव बर्थ ओनली टू द बेबी एलिफेंट एंड नॉट सम अदर एनिमल, or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents, or do they show difference in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several related questions are dealt with scientifically in the branch of the biology known as the genetics. This subject deals with the inheritance as well as the variation of the character from the parents to the offspring. Inheritance is the process by which the characters are passed on from the parent to the progeny. It is the basis of the heredity. Variation is the degree by which the progeny differ from their parents. Humans knew from as early as 8,000 BC that one of the cause of the variation was hidden in the sexual reproduction. They exploited the variation that were naturally present in the wild population of the plant and animal to selectively breed and select for the organism that possessed desirable character. For example, though artificial selection, domestication from the ancestral wild cows, we have well-known Indian breed Sahiwal cows in the Punjab. We must, however, recognize that though our ancestors knew about the inheritance of the character and the variation, they had very little idea about the scientific basis of these phenomena. तो यहाँ पर दिया हुआ है, ये दिया हुआ है सीड शेप, इधर दिया हुआ है सीड शेप है, सीड कलर है, सीड शेप में राउंड आता है, रिंकल आता है, सीड कलर में येलो और ग्रीन आता है, फ्लावर कलर में वायलेट और वाइट आता है, पॉड शेप में फुल और कंस्ट्रिक्टेड आता है, पॉड कलर में ग्रीन और येलो आता है, फ्लावर पोजीशन में एक्सेल और टर्मिनल आता है, स्टेम हाइट में टॉल और ड्वार्फ आता है. द सेवन पेयर्स ऑफ कॉन्ट्रास्टिंग ट्रेट इन द प्री प्लांट स्टडी बाय द मेंडल है जो दिए हुए. फिर आता है मेंडल्स लॉ ऑफ द इनहेरिटेंस. तो इट वाज ड्यूरिंग द मिड 19th century that headways were made in the understanding of the inheritance. Gregor Mendel conducted a hybridization experiment on the garden peach for the 7 years 1856-1863 and proposed the law of the inheritance in the living organism. During the Mendel's investigation into the inheritance patterns, it was for the first time that statistical analysis and the mathematical logic were applied to the problems in the biology. His experiment had a larger sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inference from the experiment on the successive generation of his test plant proved that this result pointed to the general rules of the inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as the two opposing traits, example tall or the dwarf plant, yellow or the green seeds. This allowed him to set up a basic framework of the rules governing the inheritance, which was expanded by the later scientists to account for the all diverse natural observation and the complexity inherent in them. फिर कहता है कि मेंडल connected the such artificial pollination cross pollination experiment using several true breeding pea lines. A true breeding line is one that having undergone uh, continuous self-pollination for the stable trait inheritance and expression for the several generation. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for the one character with contrasting trait. Some of the contrasting traits selected were smooth or wrinkled seeds, green, yellow or the green pods, inflated or the constricted green or the yellow pods on the tall or the dwarf plant. फिर आता है कॉन्ट्रास्टिंग ट्रेट स्टडी बाय द मेंडल इन द पी. तो इसमें आता है स्टेम हाइट टॉल या डॉर्फ आता है, फ्लावर कलर वायलेट वाइट आता है, फ्लावर पोजीशन एक्सियल टर्मिनल आता है, पॉड शेप इन्फ्लेटेड कॉन्स्ट्रिक्टेड आता है, पॉड कलर ग्रीन येलो हो सकता है, सीड शेप राउंड रिंकल हो सकता है और सीड कलर येलो और ग्रीन हो सकता है. तो इधर दिया हुआ है, इसमें हो गया पेटल हो गया, स्टिग्मा हो गया, एंथर हो गया, स्टेमन हो गया, कार्पल हो गया. रिमूवल ऑफ द एंथर इमैस्कुलेशन करते हैं, पेरेंट हो गया, ट्रांसफर ऑफ द पॉलिनेशन किए और स्टेप्स इन द मेकिंग ए क्रॉस इन द पी है जो दिए हुए हैं. फिर आता है इनहेरिटेंस ऑफ द वन जीन. तो Ncert कहता है कि let us take the example of the one such hybridization experiment carried out by the Mendel where he crossed tall and the dwarf pea plant to study the inheritance of the one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plant were tall like one of these parents, none were dwarfs. He made similar observation for the other pair of the trait. He found that F1 always resembled either of the parent and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that the filial two generation some of the parents were dwarf, the character that was not seen in the F1 generation by not expressed. The proportion of the plant that were dwarf were 1/4 of the F2 plant, while the 3/4 of the plant were tall. The tall and the dwarf traits were identical to their parental type and not for any blending. That is, all the offspring were either tall or the dwarf, none were of the intermediate in between height. फिर कहता है कि similar results were obtained with the other traits that he studied. Only one of the parental traits were expressed in the F1 generation, while at the F2 stage both the traits were expressed in the proportion 3:1. The contrasting trait did not show any blending at either F1 or the F2 stage. फिर कहता है based on these observations, Mendel proposed that something was being extremely passed down unchanged from the parent to the offspring through the gametes over successive generations. He called these things as the factors. Now we call them as the genes. Genes therefore are the unit of the inheritance. They contain the information that is required to express particular trait in an organism. Genes which code for a pair of the contrasting traits are known as the alleles. That means they are the slightly different form of the same gene. If we use the alphabetical symbol for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the trait expressed in the other trait. For example, in the case of the trait of the height, T is used for the tall trait and T for the dwarf. For the T and the T are the alleles of each other. Hence, in the plant, with the pair of the alleles for the height would be capital T, capital T, capital T, small t, and that small t, small t. Mendel also proposed that in a true breeding tall or dwarf pea variety, the allelic pair of the genes for the height are identical or homozygous, capital T and the small t respectively. Capital T and the small t are called the genotype of the plant, while the descriptive terms tall and the dwarf are the phenotype. But then would be the phenotype of plant and that has the genotype small t, capital T. As Mendel found that phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in the appearance, he proposed that in a pair of the dissimilar factor, one dominates the other as in the F1 and hence is called as the dominant factor while the other factor is recessive. In this case, T for the tallness is dominant over the T for the dwarfness that is recessive. He observed identical behavior for all the other characters trait pairs that he studied. It is convenient and logical to use the capital and the lowercase of an alphabetical symbol to remember this concept of the dominance and recessiveness. Please notice T for the tall and D for the dwarf because you will find it difficult to remember whether T and D are the alleles for the same gene, a character or not. Alleles can be similar as in the case of the homozygote, T, capital T, and the small t can be dissimilar as in the case of the heterozygote, small t, capital T. Since since the capital T small t plant is heterozygous for the genes controlling one character height, it is monohybrid, and the cross between the capital small t and the capital T is the monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and the dwarf plant produce gamete by the process of the meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of the allele is a random process and so there is a 50% chance of the gamete containing either allele, as verified by the result of the crossing. In this way, the gamete of the tall Tt plant have the allele T and the gamete of the dwarf Tt plant have the allele T. During fertilization of the two alleles T from the one parent phase through the pollen and T from the other parent then through the egg are united to produce the zygote that have one T allele and the one T allele. In other words, the hybrids have T. Since these hybrids contain alleles which express contrasting traits, these plants are the heterozygous. The production of the gamete by the parents, the formation of the zygote, the F1 and the F2 plant can be understood from a diagram called the Punnett square. It was developed by the British genetist Reginald C. Punnett. It is a graphical representation to calculate the probability of all the possible genotype of the offspring in a genetic cross. The possible gametes are written on the two sides, usually the top down and the left column, as possible recombination are represented in the boxes below in the square, which generate a square output form. फिर कहता है कि the Punnett square for the parental tall Tt male and the dwarf Tt female plant, the gametes produced by them are the F1 Tt progeny. The F1 plant of the genotype capital T small t are the self-pollinate. The symbols male and the female and the male are used to denote the female and the male pollen of the F1 generation respectively. The F1 plant of the genotype when self-pollinated produce gametes of the type T and T in equal proportion. When fertilization takes place, the pollen grain of between the T have 50% chance to pollinate egg for the genotype T as well as the genotype T. Also pollen grains of the genotype T have the 50% chance. तो ये दिया हुआ है इधर फिनोटाइपिक रेशियो टॉल डॉट में 3:1 और जीनोटाइपिक रेशियो दिया हुआ है 1:2:1 दिए हुए। तो ये हो गया टॉल, इधर हो गया द्वार्फ, ये हो गया फ्लिपिंग करवाएं तो इधर गैमेट है जो बन गया। इसके बाद फिर आता है कि ऑफ द pollinating egg of the genotype T as well as the genotype T as a result of the random fertilization, the resultant zygotes can be of the genotype capital T and the capital T small t or the small t small t. From the Punnett square, it is easily seen that the 1/4 of the random fertilization lead to the capital T, half lead to the capital T small t, and the 1/4 lead to the small t. Though the F1 have a genotype of the capital T small t, but the phenotypic characteristic is tall. At the F2, 3/4 of the plant are the tall, but some of them are the capital T, but the others are the capital T small t. Externally, it is not possible to distinguish between the plant with the genotype capital T and the small t capital T. Hence, for the genotypic pair capital T small t, one character T tall is expressed. Hence, the character T for the tall is said to be dominant over the other allele T or the small t or the dwarf character. It is due to the dominance of the one character over the other that all F1 are at all, though the genotype is TT, and in the F2, 3/4 of the plants are at all, though the genotypically half are TT and only 1/4 are the TT. This leads to the phenotypic ratio of the 3/4 tall, 1/4 TT and a half TT and the 1/4 TT, that means 3:1 ratio, but the genotypic ratio of 1:2:1. The 1/4:1/2:4 ratio of the TT and the TT mathematically condensable into the form of the binomial expression x plus by whole square that has a gamete bearing gene T or the T in the equal frequency of the half. The expression is expanded as given below: half T plus half T whole square, half T plus half T into half T into half T, half 4 TT, 1/4 TT or 1/4 TT. इसका मतलब आता है कि Mendel self-pollinated the F2 plant and found that the dwarf F2 plants continue to generate dwarf plants in the F3 and the F4 generation. He concluded that the genotype of the dwarf was homozygous TT. What do you think he would have got if he pollinated a tall F2 plant? इसके बाद इंसर्ट कहता है कि फ्रॉम द प्रीसीडिंग पैराग्राफ इट इज क्लियर दैट दो द जीनोटिपिक रेशियो कैन बी कैलकुलेटेड यूजिंग द मैथमेटिकल प्रोबेबिलिटी बाय सिंपल लुकिंग एट द phenotype of a dominant trait. It is not possible to know the genotypic composition. That is, for example, whether a tall plant from the F1 or F2 has TT or the TT composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed a tall plant from the F2 with a dwarf plant. This is called a test cross. In a typical test cross, an organism P plant showing a dominant phenotype and whose genotype is to be determined is crossed with a recessive parent instead of the self-crossing. The progeny of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 4.5 shows the result of the typical test cross where the violet flower is dominant over the white flower. Using the Punnett square, try to find out the nature of the offspring of a test cross. What ratio did you get? Using the genotype of this cross can give a general definition for the test cross. Based on his observation of the monohybrid cross, Mendel proposed two general rules to consolidate his understanding of the inheritance in the monohybrid cross. Today, these rules are called as the principles of the law of the inheritance, the first law of the dominance and the second law of the segregation. तो ये दिया हुआ है ये डायग्रामेटिक रिप्रेजेंटेशन ऑफ द टेस्ट क्रॉस दिया हुआ है. इधर हो गया ये होमोजाइगस रिसेसिव, ये होमोजाइगस रिसेसिव हो गया. ऑल फ्लावर्स आर वायलेट, नॉन फ्लावर फिर होमोजाइगस डोमिनेंट हो गया. फिर डोमिनेंट फिनोटाइप जीनोटाइप फ्लावर, फिर हाफ ऑफ द फ्लावर्स आर वायलेट एंड हाफ ऑफ द फ्लावर्स आर वाइट हो गया. एंड नॉन फ्लावर इज हेटेरोजाइगस हो गया. उसके बाद फिर आता है लॉ ऑफ द डोमिनेंस. लॉ ऑफ द डोमिनेंस फिर आता है तो इसमें कहता है कि फैक्टर्स आर कंट्रोल्ड बाय द डिस्क्रीट यूनिट कॉल्ड फैक्टर्स. फैक्टर्स occur in the pair. In a dissimilar pair of the factors, one member of the pair dominates or the other recessive. The law of the dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of the 3:1 obtained at the F2. Law of segregation. So this law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parent contains the two alleles during the gamete formation, the factor or the alleles of the pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all the gametes that are similar, while a heterozygous one produces two kinds of gamete, each having one allele with equal proportion. फिर दिया हुआ है result of the monohybrid cross in the parent of snapdragon where one allele is incompletely dominant over the other allele. The phenotypic ratio दिया हुआ 1:2:1 और genotypic ratio भी दिया हुआ 1:2:1 दिए हुए. तो ये हो गया F1 जनरेशन, ये हो गया F2 जनरेशन. ये ऑल पिंक हो गया, ये रेड और ये हो गया वाइट. फिर आता है इनकम्प्लीट डोमिनेंस. तो इसमें कहता है कि when experiments on peach were repeated using other traits in the other plant, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of the flower color is the dog of snapdragon or anterior is a good example to understand the incomplete dominance in a cross between the true breeding red flower and the true breeding white flower plant. And the F1 by the pink, while the F1 by itself pointed the F2 result in the following ratio 1:2:1. Here the genotypic ratio very exactly as we would expect in any Mendelian monohybrid cross, but the phenotypic ratio had changed from 3:1 dominant recessive. What happened by that R was not completely dominant over the R, and this made it possible to distinguish RR as a pink from the RR red and RR white. फिर आता है explanation of the concept of the dominance. So what exactly is dominance? Why are some alleles dominant and some recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene as a pair of the alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some change that it has undergone, about which we will read further on and in the next chapter, which modify the information that the particular allele contains. Let us take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this enzyme, the two allelic forms. Let us assume, as in the more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate. If theoretically the modified allele could be responsible for the production of the normal or less efficient enzyme, a non-functional enzyme, or the no enzyme at all. Fricata emphatic in the first case, the modified allele is equivalent to the unmodified allele. That means it will produce the same phenotype trait. That means result in the formation of the subtrait S. Such equivalent allele pair are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to the non-functional enzyme or because no enzyme is produced. इसके बाद कहता है co-dominance. तो till now we are discussing cross with that pair. The F1 resemble either of the two parents, dominance or in between incomplete dominance. But in the case of the co-dominance, the F1 generation resemble both parent. A good example is different type of the red blood cells in that determines ABO blood grouping in the human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells is a sugar polymer that protrudes from its surface, and the kind of the sugar is controlled by this gene. The gene I has the three alleles IA, IB, and I. These alleles IA and IB produce a slightly different form of the sugar, while allele I do not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three genes IA and IB are completely dominant over the I. In other words, I and I are present only I express because I do not produce any sugar. When I and IB are present, IB expresses. But when I and IB are present together, they both express their own type of the sugar. This is because of the codominance. Hence, red blood cells have both A and the B type of the sugar. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible, and therefore total 6 different genotype of the human ABO blood types. How many phenotypes are the possible? ये पूछ रहा है. तो ये ह्यूमन का हो गया. अगर है कि आइए आइए है तो आइए आइए बनेगा तो ये बनेगा. आइए आई बी है तो आइए ए बी बनेगा, ये भी बन जाएगा. आइए आई है तो आइए आई ए बी ए बन जाएगा. फिर आई बी आइए है तो आई बी बन जाएगा. आई बी है तो आई बी ए बी बन जाएगा. फिर आई है तो आई बी बन जाएगा. और आई है तो आई ओ, फिर इसका हो जाएगा ओ बन जाएगा. फिर कहता है कि डू रियलाइज दैट एग्जाम्पल ऑफ द एबीओ ब्लड ग्रुपिंग also provides a good example of the multiple alleles. Have you seen that there are more than two? That means, have you seen that there are more than two? That means three alleles governing the same character. Since in an individual only two alleles can be present, multiple alleles can be found only when the population studies are made. Occasionally, a single gene product may produce more than one effect. For example, a starch synthesis in the pea seed is controlled by one gene. It has two alleles, B and the B. Starch is synthesized effectively by the B, capital B, homozygotes, and therefore starch grains are produced. In contrast, small b homozygotes have the lesser efficiency in the starch synthesis and produce a smaller starchy grain. After maturation of the seeds, capital B seeds are the round and the small b seeds are the wrinkled. Heterozygotes produce round seeds, and the so beam seems to be dominant allele, but the starch grain produced are of the intermediate size in the capital B small b seeds. So if the starch grain size is considered as the phenotype, then from this angle the alleles for the incomplete dominance. Therefore, the dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype. From this product, I die on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene. फिर आता है inheritance of the two gene. तो Mendel also worked with the and crossed pea plants that differ in the two character, as shown in the cross between a pea plant that has seed with yellow color and round shape and one that has seed of the green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and the round shaped seeds. Here, can you tell which of the character in the traits pair yellow green color and the round wrinkled seed by dominant? Thus, the yellow color by dominant over the green and the round shape dominant over the wrinkled. These results were identical to those that he got when he made separate monohybrid cross between the yellow and the green seeded plant and between the round and the wrinkled seeded plant. Let us see the genotypic symbol Y for the dominant seed color and Y for the recessive green seed color, R for the round shaped seed and R for the wrinkled seed shape. The genotype of the parent can be written as the RR and the RR. The cross between the two can be written down as in the figure 4.7, showing the genotype of the parent plant. The gamete RY and the RY unite on the fertilization to produce the F1 hybrid RRY. When Mendel fertilized the F1 parent, he found that 3/4 of the F2 plants had yellow seeds and the 1/4 had the green. The yellow and the green seeds segregated in a ratio 3:1. The round and the wrinkled seeds also segregated in ratio 3:1, just like the monohybrid cross. तो पेरेंटल जनरेशन है, गैमेट्स बनाया, F1 जनरेशन आ गया, फिर ये हो गया F2 जनरेशन। राउंड येलो राउंड ग्रीन, रिंकल्ड येलो और रिंकल्ड ग्रीन का रेशियो है 9:3:3:1। फिर रिजल्ट ऑफ a dihybrid cross where the two parents differ in the two pairs of the confronting trait, feed colors and the feed shape में। इसके बाद आता है लॉ ऑफ द इंडिपेंडेंट असॉर्टमेंट। तो कहता है कि इन डाइहाइब्रिड क्रॉस द फिनोटाइप राउंड येलो रिंकल्ड येलो राउंड ग्रीन and the wrinkled green appeared in the ratio 9:3:1. Such a ratio was observed for the several pairs of the characters that Mendel studied. The ratio 9:3:1 can be derived as a combination of the series of the 3 yellow, 1 green with 3 round, 1 wrinkled. The derivation can be written as the follow: 3 round, 1 wrinkled, 3 yellow, 1 green is equal to 9 round yellow, 3 wrinkled yellow, 3 round 1 green or 1 wrinkled green. Based upon such observation on a dihybrid cross between the plants differing in two traits, Mendel proposed the second set of the generalization that we call the Mendel's law of the independent assortment. The law states that when the two pairs of traits are combined in a hybrid, the segregation of the one pair of the character independent of the other pair of the character. The Punnett square can be effectively used to understand the independent segregation of the two pair of the genes during the meiosis and the production of the eggs and the pollen in the F1 RR plant. Consider the segregation of the one pair of the gene R and the R. 50% of the gamete have the gene R and the other 50% have the R. Now besides each gamete having either R or the R, it should also have the allele bi or the bi. The important thing to remember here is that segregation of the 50% of the R and the 50% of and the 50% R is independent from the segregation of the 50% of the bi and the 50% bi. Therefore, the 50% of the R bearing gametes and has the bi and the other 50% has the bi. Similarly, 50% of the R bearing gametes have bi and the other 50% has the bi. Thus, there are 4 genotype of the gamete, 4 type of the pollen and the 4 type of the eggs. The 4 type of the RR, R by, R by, R by and the R by each with a frequency of the 25% or the 1/4 of the total gamete produced. When we write down the 4 type of the eggs and pollen on the two sides of a Punnett square, it is very easy to derive the composition of the zygotes that give rise to the F2 plant. Although there are 15 squares, how many different type of the genotype and the phenotype are formed, note them down in the format given. Can we, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also 9:3:3:1? फिर इसके बाद आता है क्रोमोसोमल थ्योरी ऑफ द इनहेरिटेंस। तो कहता है मेंडल proliferate his work on the inheritance of the character in 1855, but for the several reasons it remained unrecognized till the 1900. Firstly, क्या था कि communication was not easy as it is now in those days and his work could not be widely publicized. Secondly, his concept of the genes for the factor in the Mendel bird as a stable and discrete units that controlled the expression of the traits and of the pair of the alleles which did not blend with each other by not affected by the contemporary as an explanation for the apparently continuous variation seen in the nature. Thirdly, Mendel's approach to using the mathematics to explain the biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors were discrete units, he could not provide any physical proof for the existence of the factor or say what they were made of. In the 1903 scientists debriefed Korean from the Tesmark, independently rediscovered Mendel's result on the inheritance of the character. Also by this time, due to advancement in the microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of the structure in the nucleus that appeared to be double and divided just before each cell division. They were called the chromosome-colored body as they were visualized by the staining. By 1902, the chromosome movement during the meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of the chromosome was partial due to the behavior of the genes and used the chromosome movement to explain the Mendel's law. Recall that you studied the behavior of the chromosome during the mitosis, equational division, and during the meiosis, reductional division. The important thing to remember that the chromosome as well as the genes occur in a pair. The two alleles of a gene pair are located at the homologous sites on the homologous chromosome. इधर दिया हुआ meiosis and the germ cell formation in the cell with a 4 chromosome. Can you see how chromosomes segregate when the germ cells are formed? इधर G1 है, G2 है, meiosis 1 एनाफेज हो गया, meiosis 2 एनाफेज का हो गया, इधर हो गया germ cell हो गया। तो a comparison दिया हुआ है, a comparison between the behavior of the chromosome and genes. तो occur in pair होता है, ये भी occur in pair होता है। segregate at the time of the gamete formation such that only one of each pair is transmitted to a gamete. If you segregate at the time of gamete formation, only one of the pair is transmitted to the gamete. इसमें independent pair segregate independently of each other. इसमें भी one pair segregate independently of each other. फिर कहता है during the anaphase of the meiosis one, the two chromosome pair can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of the four different colors in the left and the right columns. In the left column, possibility 1, orange and the green is segregating together, but in the right hand column, possibility 2, the orange chromosome is segregating with the red chromosome. तो बोलता है पॉसिबिलिटी 1 में कि वन लॉन्ग ऑरेंज एंड द शॉर्ट ग्रीन क्रोमोजोम एंड द वन लॉन्ग येलो एंड द शॉर्ट क्रोमोजोम एट द पोल. पॉसिबिलिटी 2, वन लॉन्ग ऑरेंज एंड द शॉर्ट रेड क्रोमोजोम एंड द शॉर्ट ग्रीन क्रोमोजोम एट द सेम पोल. तो ये मेयोसिस 1 हो गया, मेयोसिस 1 एनाफेज हो गया, मेयोसिस 2 एनाफेज हो गया, ये मेयोसिस 2 भी एनाफेज का हो गया. ये जर्म सेल हो गया, ये भी जर्म सेल हो गया. इंडिपेंडेंट असॉर्टमेंट ऑफ द क्रोमोजोम हो गया. फिर कहता है कि Sutton and the Bovary argued that the pairing and the separation of a pair of the chromosome would lead to the segregation of a pair of the factor they carried. Sutton united the knowledge of the chromosomal segregation with the Mendelian principle and called it the chromosomal theory of the inheritance. Following the synthesis of the idea, experimental verification of the chromosomal theory of the inheritance by Thomas Hunt Morgan and his colleague led to the discovering of the basis of the variation that the sexual reproduction produced. Morgan worked with tiny fruit fly Drosophila melanogaster, which were found very suitable for the such studies. They could be grown on a simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of the progeny flies. Also, there was a clear differentiation of the sexes. The male and the female flies are really distinguishable. Also, it has many types of the hereditary variation that can be seen with the low power microscope. इधर दिया हुआ है ड्रोसोफिला मिलानोगास्टर और मेल और ये मेल हो गया और ये हो गया फीमेल हो गया. ये मेल है और ये है जो फीमेल है. फीमेल जो है वो बड़े साइज का है. इसके बाद आता है लिंकेज एंड रिकॉम्बिनेशन. तो Morgan carried out several dihybrid crosses in the Drosophila to study genes that were sex-linked. The cross were similar to the dihybrid cross carried out by the Mendel in the peach. For example, Morgan hybridized yellow-bodied, white-eyed female to the brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the two genes would not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3 to 3:1 ratio expected from the two genes are the independent. Morgan and his group knew that genes were located on the X chromosome and found quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of the parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or the linkage of the two genes and coined the term linkage to describe the physical association of the genes on a chromosome and the term recombination to describe the generation of the non-parental gene combination. Morgan and his group also found that even when the genes were grouped on the same chromosome, some genes were tightly linked for very low combination, while others were really linked for higher combination. For example, he found that genes white and the yellow were highly tightly linked and showed only 1.3% recombination, while the white and the miniature being showed only 37.2% recombination. His student Alfred Student used the frequency of the recombination between the gene pairs on the same chromosome as a measure of the distance between the gene and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequence of the whole genome as by done in the case of the human genome sequencing project described later. तो जब ये है पेरेंटल हो गया, येलो वाइट यानी कि येलो बॉडीड वाइट आई को बाइल टाइप से किए हैं तो F1 जनरेशन. तो जब आई और बॉडी कलर के कर रहे हैं तो पेरेंटल आई में और बॉडी कलर के कर रहे हैं तो पेरेंटल उसमें आ रहा है 98.1% और रिकॉम्बिनेंट आ रहा है 31.3%. और जब आई वाइट और मिनिएचर यानी आई और बिंग को कर रहे हैं तो पेरेंटल 52.8% आ रहा है और रिकॉम्बिनेंट आ रहा है 37.2%. कब? जब आई को और बिंग को लेकर कर रहे हैं तब. तो linkage result of the two dihybrid cross conducted by the Morgan, cross F for the crossing between the gene Y and the W, cross B for the crossing between the W and the M. Her dominant bitype alleles are represented with the plus sign in the superscript. The strength of the linkage between Y and W is higher than the W and M. फिर आता है पॉलीजेनिक इनहेरिटेंस. तो मेंडल कहता है कि Mendel's studies mainly describe those traits that have been distinct alternate forms such as the flower color which were either purple or white. But if you look around, you will find that there are many other traits which are not so distinct in their occurrence and spread across the cadence. For example, in the humans, we don't just have tail or the short people as the two distinct alternatives, but a whole range of the possible height. Such traits are generally controlled by the three or the more genes, and others are called as the polygenic traits. Besides the involvement of the multiple genes, polygenic inheritance also take place into account the influence of the environment. Human skin color is another classic example of for this. In the polygenic trait, the phenotype reflects the contribution of the each allele. That means the effect of the each allele is additive. To understand this better, let us assume that three genes ABC control a skin color in the human, with the dominant form ABC responsible for the dark skin color and the recessive form ABC for the light skin color. The genotype with all the dominant allele will have the darkest skin color, and that with the recessive allele will have the lightest skin color. As expected, the genotype with the three dominant allele and the three recessive allele will have intermediate skin color. In this manner, the number of each type of the alleles in the genotype would determine the darkness or the lightness of the skin in an individual. फिर आता है pleiotropy. So we have been so far seen the effect of the gene on a single phenotype or the trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. The underlying mechanism of the pleiotropy in the most cases is the effect of the gene or the metabolic pathway which contribute towards the different phenotype. An example of this is the disease phenylketonuria which occurs in the humans. The disease is caused by the mutation in the gene that codes for the enzyme phenylalanine hydroxylase, single gene mutation. This manifests itself through the phenotypic expression characterized by the mental retardation and a reduction in the hair and the skin pigmentation. इसके बाद फिर आता है सेक्स डिटर्मिनेशन. द मैकेनिज्म ऑफ द सेक्स डिटर्मिनेशन हैज ऑलवेज बीन अ पजल बिफोर द जेनेटिस्ट. द इनिशियल clue about the genetic chromosomal mechanism of the sex determination can be tracked back to some of the experiments carried out in the insect. In fact, the phytological observation made in a number of the insects led to the development of the concept of the genetic chromosomal basis of the sex determination. Hanging at Africanum could trace a specific nuclear structure of all through the spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure and after spermatogenesis, whereas the other 50% did not receive it. Hanking gave a name to this structure as the X body, but he could not explain its significance. Further investigation by the other scientists led to the conclusion that the X body after Hanking was in fact the chromosome, and that why it was given the name X chromosome. It was observed that in the large number of the insects, the mechanism of the sex determination is of the exotype. That means all the X bear an additional X chromosome besides the other chromosome autosomes. On the other hand, some of the sperm bear the X chromosome, whereas some do not. Eggs fertilized by the sperm bearing an X chromosome becomes females, and those fertilized by the sperm that do not an X chromosome becomes male. Do you think the number of the chromosome in the male and the female are equal? Due to involvement of the X chromosome in the determination of the sex, it was designated to the sex chromosome, and the rest of the chromosome were named as the autosomes. Graph upper is an example of the exotype of the sex determination in which the males have only one X chromosome besides the autosomes, where the female have a pair of the X chromosome. फिर कहता है कि the observation led to the investigation of a number of the species to understand the mechanism of the sex determination in a number of the other insects and mammals, including the man. XY type of the sex determination is seen where both male and the female have the same number of the chromosomes. Among the male, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Female, however, have a pair of the X chromosome. Both male and the female bear the same number of the autosomes. Hence, the male have autosomes plus XY, while the female have autosomes plus XX. In the human being and in Drosophila, the male have the one X and the one Y chromosome, whereas the female have one pair of the X chromosome besides the autosomes. In the above description, we have studied about the two types of the sex determining mechanism, that means XO type and XY type. But in both cases, male produce two types of the two different type of gamete, either with or without the X chromosome, or some gametes with the X chromosome and some with Y chromosome. Such type of sex determination mechanism is designated to be the example of the male heterogamety. In some other organism, example birds, a different mechanism of the sex determination is observed. In this case, the total number of the chromosomes is same in both males and females, but the two different types of the gametes in the term of the sex. chromosome are produced by the female. Female heterogamety, in order to have a distinction with the mechanism of the sex determination described earlier, the two different sex chromosomes of a male female bird have been reunited to be the Z and W chromosome in this organism. The female have one Z and one W chromosome, whereas the male have a pair of the Z chromosome besides the autosomes. तो इधर दिया हुआ है डिटर्मिनेशन ऑफ, इधर दिया हुआ है क्या दिया हुआ है, तो ये है डिटर्मिनेशन ऑफ दी, ये दिया हुआ है determination of the sex by the chromosome differences both in the humans and in a Drosophila. The female has a pair of the XX chromosome homogametic and the male X by heterogametic composition. In many birds, female has a pair of the dissimilar chromosome ZW and the male two dissimilar ZZ chromosome दिया हुआ है. तो ये हो गया मेल का, ये हो गया फीमेल का, ये हो गया एक्स वाई और इसमें भी एक्स एक्स यानी कि सेम एज इसके तरह ही दिए हुए. फिर जेड जेड हो गया, इधर आ गया जेड डब्ल्यू टाइप का होता है बर्ड्स में, यानी कि बर्ड्स में जो मेल हो गया वही है जो होमोगेमेटिक है. फिर आता है सेक्स डिटर्मिनेशन इन द ह्यूमंस में कैसे होता है. तो इट हैज ऑलरेडी बीन मेंशन दैट सेक्स डिटरमाइनिंग mechanism in the case of the human is the XY type. Out of the 23 pair of the chromosome present, 22 pairs are exactly same in both the males and the females. These are the autosomes. A pair of the X chromosome are present in the female, whereas the presence of an X and Y chromosomes are determinant of the male characteristic. During the spermatogenesis among males, two types of these gametes are produced. 50% of the total sperm produced carry the X chromosome and the rest 50% carry the Y chromosome besides the autosomes. Female, however, produce only one type of the ovum with the X chromosome. There is an equal probability of the fertilization of the ovum with the sperm carrying either X or the Y chromosome. In case the ovum fertilized with a sperm carrying X chromosome, the zygote develops into a female XX and the fertilization of the ovum with the Y chromosome carrying a sperm result into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female bird. It is unfortunate that in our society women are blamed for giving birth to the female children and have been ostracized and ill-treated because of this false notion. तो उसके बाद आता है सेक्स डिटर्मिनेशन इन द हनी बी में. तो the sex determination in the honeybee is based on the number of the sets of the chromosome and individual received. An offspring formed from the union of a sperm and the egg develop a female queen or worker, and an unfertilized egg develops a male by the means of the parthenogenesis. This means that the males have half the number of the chromosome than that of a female. The females are diploid having 32 chromosomes and the males are haploid, that means having the 16 chromosomes. This is called as the haplodiploid sex determination system and a special characteristic features such as the male produce of sperm by the mitosis do not have father and thus cannot have sons but have the grandfather and can have the grandsons. How the sex determination mechanism produced in the birds is the sperm or the egg responsible for the sex of the chicks. इधर दिया पेरेंट का, ये गैमेट्स का, ये स्पर्म का. इधर सेक्स डिटर्मिनेशन इन द हनी बी में दिया हुआ है. फिर आता है म्यूटेशन. तो म्यूटेशन के बारे में है, एनसीईआरटी लिखता है कि म्यूटेशन इज ए फिनोमेना व्हिच रिजल्ट्स इन द अल्टरेशन ऑफ द डीएनए सीक्वेंस एंड consequently results in the change in the genotype and the phenotype of an organism. In addition to the recombination, mutation is another phenomena that leads to the variation in the DNA. As you will learn in the chapter 5, one DNA helix runs continuously from the one end to the other in each chromatid is highly supercoiled from, therefore loss, a deletion, or gain, insertion, a duplication of a segment of the DNA result in the alternation in the chromosome. Since genes are known to be located on the chromosome, alternation in the chromosome results in the abnormalities or the aberration. Chromosomal aberrations are commonly observed in the cancer cells. In addition to the above, mutation also arise due to the change in a single base pair of the DNA. This is known as the point mutation. A classical example of such a mutation is the sickle cell anemia. Deletion and insertion of the base pair of the DNA cause the frame shift mutation. The mechanism of the mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutation. These are referred to as the mutagens. UV radiations can cause the mutations in an organism. It is a mutagen होता है. फिर इधर दिया हुआ है, ये हो गया पेडिग्री, ये हो गया पेडिग्री एनालिसिस. जेनेटिक डिसऑर्डर में पेडिग्री एनालिसिस का देखते हैं. तो ये पेडिग्री एनालिसिस हो गया. द आइडिया that the disorders are inherited has been prevailing in the human society since long. This was based on the heritability of the certain characteristic feature in the families. After the rediscovery of the Mendel work, the practice of the analyzing inheritance pattern of the traits in the human beings began, since it is evident that control crossage that can be performed in a pea plant or some other organism are not possible in the case of the human being. A study of the family history about inheritance of a particular trait provides an alternative. Such analysis of the trait in the several generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a human trait of a particular trait is represented in a family tree over the generation. In the human genetics, predictive study provides a strong tool which is utilized to trace the inheritance of the specific trait, abnormality, or the disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 4.13. As you studied in this chapter, each and every feature of an organism controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of the genetic information. It is hence transmitted from the one generation to the other without any change or alternation. However, changes or alternation do take place occasionally. Such an alternation or changes in the genetic material is referred to as the mutation. A number of the disorders in the human beings have been found to be associated with the inheritance of the changed or the altered genes or the chromosomes. तो ये दिखाया हुआ सिंबल यूज इन द ह्यूमन पेडिग्री एनालिसिस में. तो स्क्वायर वाला मेल का हो गया, राउंड वाला फीमेल का हो गया, इसका सेक्स अनस्पेसिफाइड या अफेक्टेड इंडिविजुअल्स हो गया. मेटिंग दिखा रहा है, एक मेटिंग बिटवीन रिलेटिव कॉन्सिन्यूस मेटिंग दिखा रहा है. पेरेंट्स हम चाइल्ड इन ऑर्डर टू गिव द बर्थ लेफ्ट टू द राइट. पेरेंट्स विद द मेल चाइल्ड अफेक्टेड विथ डिवीज द्वारा फाइव अफेक्टेड ऑफ स्प्रिंग का दिखा रहा है. इसके बाद फिर आता है मेंडेलियन डिसऑर्डर. तो broadly genetic disorder may be grouped into two categories: Mendelian disorder and chromosomal disorder. Mendelian disorder are mainly determined by the alternation or the mutation in the single gene. These disorders are transmitted to the offspring on the same line as we have studied in the principle of the inheritance. The pattern of the inheritance of such Mendelian disorder can be traced in a family by the pedigree analysis. Most common and the prevalent Mendelian disorder are the hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorder may be dominant or the recessive. By the pedigree analysis, one can easily understand whether the trait in the question is dominant or recessive. Similarly, the trait may also be linked to the X chromosome, as in the case of the hemophilia. It is evident that the X-linked recessive trait for the transmission from the carrier female to the male progeny. A representative pedigree is shown in figure 4.14 for the dominant and the recessive trait. Discuss with your teacher and design pedigree for the character linked to the both autosome and the sex chromosome. तो यहाँ पर दिखाया हुआ रिप्रेजेंटेशन पेडिग्री एनालिसिस ऑटोसोमल डोमिनेंट ट्रेट फॉर दी मायोटोनिक डिस्ट्रॉफी एंड ऑटोसोमल रिसेसिव ट्रेट फॉर दी सिकल सेल एनीमिया के लिए दिखाया हुआ है. ये सिकल सेल एनीमिया है, यानी कि ये हो गया ऑटोसोमल रिसेसिव हो गया और ये हो गया मायोटोनिक डिस्ट्रॉफी ऑटोसोमल डोमिनेंट. कैसे पता करेंगे कि अगर ऑटोसोमल में अगर पेरेंट्स अफेक्टेड नहीं है लेकिन प्रोजेनी अफेक्टेड है तो ये ऑटोसोमल रिसेसिव है. पेरेंट्स अफेक्टेड नहीं है लेकिन प्रोजेनी अफेक्टेड है तो ये ऑटोसोमल रिसेसिव हो गया. इसमें क्या होता है कि इसमें ऑटोसोमल डोमिनेंट में क्या होता है कि इसमें है जो ऑटोसोमल डोमिनेंट में अगर तुम्हारा ये फीमेल है जो अफेक्टेड है तो चाइल्ड भी अफेक्टेड हो जाएगा. तो सन अफेक्टेड होगा तो ये जो ऑटोसोमल डोमिनेंट होता है. फिर आता है कलर ब्लाइंडनेस. तो कलर ब्लाइंडनेस में क्या आता है, तो कलर ब्लाइंडनेस भी सेक्स लिंक्ड है. तो इट इज सेक्स लिंक्ड रिसेसिव डिसऑर्डर ड्यू टू द defect in the either red or the green cone of the eye, resulting in the failure to discriminate between the red and the green color. This defect is due to the mutation in the certain gene present in the X chromosome. It occurs in about 8% of the males and only about 0.4% of the females. This is because the genes that lead to the red-green color blindness are on the X chromosome. Males have only one X chromosome and the females have two. The sons of the woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. This means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is carrier and her father is colorblind. फिर आता है हीमोफीलिया. This sex-linked recessive disease which for its transmission from the unaffected carrier female to the sum of the male progeny has been widely studied. In this disease, a single protein that is a part of the cascade protein involved in the clotting of the blood is affected. Due to this, in an affected individual, a simple cut will result in a non-stop bleeding. The heterozygous female for hemophilia may transmit the disease to the son. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic. Unviable in the later stage of your life. The family pedigree of the Queen bacteria for a number of the hemophilic descendants achieved by the carrier of the disease. There are the sickle cell anemia. This is an autosome linked recessive trait that can be transmitted from the parent to the offspring when both the partners and are the carriers for the gene or the heterozygous. The disease is controlled by a single pair of the allele HBA and HBF. Out of the three possible genotypes, only homozygous individual for the HBF for the disease phenotype. A heterozygous HBA-HBF individual appear apparently unaffected, but they are the carrier of the disease at the 50% probability of the transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The effect is the defect is caused by the substitution of the glutamic acid by the valine at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of the amino acid in the globin protein results due to the single base substitution at the sixth codon of the beta globin gene from the GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RNA from the biconcave disc to elongated sickle cell structure. ये यहाँ पर दिया हुआ है micrograph of the red blood cells and the amino acid composition of the relevant portion of the beta chain of the hemoglobin from a normal individual from an individual with the sickle cell anemia के लिए यहाँ पर दिए हुए. फिर उसके बाद आता है फिनाइल कीटोनिया. तो दिस इंसर्ट कहता है कि दिस इनबॉर्न error of the metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in the brain results in the mental retardation. They are also excreted through the urine because of its poor absorption by the kidney. फिर आता है थैलेसेमिया. तो दिस is also an autosome linked recessive blood disease transmitted from the parent to the offspring when both the parents are unaffected carrier of the for the gene. The defect could be due to either mutation or deletion, which ultimately leads to the reduced rate of the synthesis of one of the globin chain, alpha and the beta chains that make up the hemoglobin. This causes the formation of the abnormal hemoglobin molecule, resulting into the anemia, which is characteristic for the after disease. Thalassemia can be classified according to which the chain of the hemoglobin molecule is affected. In alpha thalassemia, production of the alpha globin chain is affected, while in the beta thalassemia, production of the beta globin chain is affected. Alpha thalassemia is controlled by the two closely linked genes HBA1 and HBA2 on the chromosome 15 of the each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecule produced, while the beta thalassemia is controlled by a single gene HBB on the chromosome 11 of the each parent and occurs due to mutation of the one or both of the genes. Thalassemia differs from the sickle cell anemia in that the former is quantitative problem of the synthesizing too few globin molecules, while the latter is the qualitative problem of the synthesizing and incorrectly functioning globin. इसके बाद फिर आता है क्रोमोजोमल डिसऑर्डर. तो क्रोमोजोमल डिसऑर्डर में the chromosomal disorder, on the other hand, are caused due to the absence or the excess of the abnormal arrangement of one or more chromosome. Failure of the segregation of the chromatids during the cell division cycle results in the gain or loss of the chromosome called aneuploidy. For example, Down syndrome results in the gain of the extra copy of the chromosome 21. Similarly, Turner syndrome results due to the loss of an X chromosome in the human females. Failure of the cytokinesis of the telophase stage of the cell division results in increase in the whole set of the chromosome in an organism, and this phenomenon is the polyploidy. This condition is often seen in the plants. The total number of the chromosomes in a normal human cell is 46. 23 pairs out of these 22 pairs are autosomes, and one pair of the chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of the chromosome may be included in an individual, or an individual may lack any one of the pair of the chromosome. These situations are known as the trisomy or the monosomy of a chromosome respectively. Such a situation leads to the very serious consequences in the individuals. Down syndrome, Turner syndrome, Klinefelter syndrome are common example of the chromosomal disorder. फिर यहाँ पर दिया हुआ alternative figure showing an individual inflicted with the Down syndrome and the corresponding chromosome of the individual. ये black flat back of the head है, many loops on the fingertips है, palm crease है, broad flat face है, big and the wrinkled tongue है, condenser heart disease हो गया. फिर यहाँ पर दिया हुआ है tall of stature with feminine character, इधर short of stature with underdeveloped feminine character दिया हुआ है. फिर आता है Down syndrome. That the cause of this genetic disorder is the presence of an additional copy of the chromosome 21, trisomy 21. This disorder was first discovered by the Langdon Down. The affected individuals is short of stature with a small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm crease, physical psychomotor, and the mental development is retarded. फिर आता है clean fetal freedom. So this genetic disorder is also caused due to the presence of an additional copy of the X chromosome resulting into a karyotype of the 47 XX. By such an individual has overall masculine development. However, the feminine development of the breast gynecomastia is also expressed. Such individuals are sterile. So yeah, it had the diagrammatic representation of the genetic disorder due to the female sex chromosome in the humans. ये दिया हुआ है क्लाइन-फोस्टर सिंड्रोम और ये दिया हुआ है टर्नर सिंड्रोम का ये दिया हुआ है. फिर आता है टर्नर सिंड्रोम. तो आता है such a disorder is caused due to the absence of the X chromosome, that means 45 with the X4. Such females are sterile as ovaries are rudimentary besides other features including lack of the other secondary sexual characters. तो फिगर दिए हुए. अब इसका समरी आता है. तो समरी है कि genetics is the branch of the biology which deals with the principle of the inheritance and its practices. Progeny resembling the parents in the morphological and the physiological feature has attracted the attention of the many biologists. Mendel was the first to study this phenomena systematically while studying the pattern of the inheritance in the pea plant of the contrasting character. Mendel proposed the principle of the inheritance which are today referred to as the Mendel's law of the inheritance. He proposed that the factors later named the genes regulating the characters are found in the pairs known as alleles. He observed that the expression of the character in the offspring follow a definite pattern in the different first generation, second, and so on. Some characters are dominant over others. The dominant characters are expressed when factors are in heterozygous condition, law of dominance. The recessive characters are only expressed in the homozygous condition. The characters never blend in the heterozygous condition. A recessive character that was not expressed in the heterozygous condition may be expressed again when it becomes homozygous. Hence, characters segregate by the formation of the gametes, law of segregation. फिर कहता है कि not all the characters for the true dominance, some characters for the incomplete and some for the co-dominance. When Mendel studied the inheritance of the two characters together, it was found that the factors independently assort and combine in all the permutations and the combinations of the independent assortment. Different combinations of the gametes were theoretically represented in a square tabular form known as the Punnett square. The factors now known as genes on the chromosomes regulating the characters are called the genotype and the physical expression of the character is called as the phenotype. Figure after knowing that the genes are located on the chromosome A, good correlation was drawn between the Mendel's law segregation and the assortment of the chromosome during meiosis. The Mendel's law were extended in the form of the chromosomal theory of the inheritance. Later, it by found that Mendel's law of independent assortment did not hold true for the genes that were located on the same chromosome. These genes were called as the linked genes. Closely located genes assorted together and differently located genes due to recombination assorted independently. Linkage maps therefore correspond to the arrangement of the genes on a chromosome. फिर एंसर कहता है कि many genes were linked to the sexes also and also called as the sex linked genes. The two sexes, male and female, were found to have the set of the chromosome which were common and other set which was different. The chromosome which were different in the two sexes were named as the sex chromosome. The remaining set were named as the autosomes. In human, a normal female has 22 pairs of the autosomes and a pair of the sex chromosome. A male has 22 pairs of the autosomes and a pair of the sex chromosome. In chicken, sex chromosome in male are the ZZ and in the female are the ZW. फिर कहता है कि mutation is defined as a change in the genetic material. A point mutation is a change of a single base pair in the DNA. Sickle cell anemia is caused due to the change of one of the base in the gene coding for the beta chain of the hemoglobin. Inheritable mutation can be studied by generating a pedigree of a family. Some mutations involve change in the whole set of the chromosome polyploidy or change in the subset of the chromosome number aneuploidy. This helps in understanding the mutation basis of the genetic disorder. Down syndrome is due to the trisomy of the chromosome 21, where there is an extra copy of the chromosome 21 and consequently the total number of the chromosome becomes 47. In the Turner syndrome, the one X chromosome is missing and the sex chromosome is an X4, and in the Klinefelter syndrome, the condition is the XXY. These can be illustrated by the analysis of the karyotypes. ये हो गया जेनेटिक्स.